Hello students, and welcome to your second lecture on hematology. I'm Dr. Kasonga. Again, my office is BMS 912. If you have any questions, please feel free to visit me in my office, or you can email me on my email address over here. In this lecture, we will discuss haematopoiesis. Now, haematopoiesis is the process by which your blood cells are formed. So what is haematopoiesis? As I said, it's the process by which your blood cells are formed. Your red blood cells and your granulocytes are mostly produced in your red bone marrow, whereas your agranulocytes are mostly produced in your spleen, your thymus, or your lymph nodes. If you remember from your previous lecture, your agranulocytes are your monocytes and your lymphocytes, and they do not have granules in their cytoplasm. Your granulocytes are your neutrophils, your eosinophils, and your basophils, and they do have granules in their cytoplasm. And then your other cellular elements or your other formed elements in your blood are your erythrocytes or your red blood cells and your platelets. So the site of blood cell production can differ depending on your stage of life. As a fetus, in the mesoblastic phase, your blood cells are produced in the yolk sac. In the hepatic phase, that's between 3 to 6 months, your liver will take over as the primary site of your blood cell production. And there is some contribution from your spleen, your lymph nodes, and your thymus as well. In the myeloid phase, so that's at 6 months, the red bone marrow will take over as the primary site of your blood cell production. In adolescence, your bone marrow is the primary site, and it's primarily in the marrow of your sternum, your ribs, your skull, your vertebra, the pelvis, the femur, and the tibia. As you enter adulthood, some of these sites will cease to produce blood cells. So it'll only be the sternum, the ribs, the skull, the vertebra, and the pelvis that will be the primary sites of your blood cell production. During abnormal circumstances, the liver and the spleen can resume producing blood cells, and this is known as extramedullary haematopoiesis. And this is typically a sign of a pathological condition that has caused the destruction of your bone marrow, and that's why these other sites will then have to take over to produce your blood cells. So this is a longitudinal section of a long bone, and you can see in this upper portion of your bone you have this porous bone. You can see there's a lot of gaps in the bone, and this is known as the spongy bone. And in these pores in your spongy bone, this is where you have your red bone marrow. So in your red bone marrow, this is where you produce your blood cells. This will be the primary site of blood cell production during adulthood. In the middle of your bone over here, you will have your yellow marrow. And in your yellow marrow, you'll form your mesenchymal stem cells, which are important for the formation of cartilage, fat, and bone. The bone marrow is quite a substantially sized tissue. If you had to collect all the bone marrow from the bones, it would be about the same size and weight as the liver. And the liver is one of the largest organs in the body, so it tells you that this is quite a substantially sized tissue. In total, the bone marrow makes up about 4% of your body weight. Your bone marrow is also a highly vascular tissue, so there's a lot of blood vessels that enter into this tissue. And this is important because your bone marrow requires a lot of energy in order to produce new blood cells every day. You make about 500 billion cells in your bone marrow every single day. So it's a very metabolically active tissue, and it needs a lot of oxygen and a lot of energy in order to make all these cells. Your cells also have to be able to readily enter into your circulation. So because you're forming blood cells, they have to be able to get into the blood very easily. And that's another reason why it's a highly vascular tissue. The bone marrow is filled with blood cells at varying stages of development. Here you can see a megakaryocyte that is forming your platelets. There's a red blood cell stem cell that's forming your red blood cells, and a neutrophil stem cell that's forming your neutrophils. About 75% of the developing cells in your marrow will become white blood cells, and the other 25% will become your red blood cells. Now this is because red blood cells have a longer lifespan than white blood cells. Apart from your memory lymphocytes, Your white blood cells typically have a lifespan of between a few hours and a few days, whereas your red blood cells can last for as long as 120 days. So therefore your white blood cells have to be replenished more frequently than your red blood cells. So all red blood cells will start off as a multipotent stem cell. And this multipotent stem cell can either differentiate into a myeloid progenitor or a lymphoid progenitor, depending on the signals that it receives. This myeloid progenitor can become a megakaryocyte, which will form your platelets. It can also become an erythroblast, which will eventually form your red blood cells, or a myeloblast, which will form your monocytes, your neutrophils, your basophils, and your eosinophils. Your lymphoid progenitor is what will form your lymphocytes. These include your B lymphocytes, your T lymphocytes, and your natural killer cells. So let's take a look at some of these important factors in the stimulation of your blood cell production. So for your red blood cells, EPO or erythropoietin is the most important factor in the production of your red blood cells. EPO is produced by your kidney cells and it stimulates the production of more red blood cells. Then you have thrombopoietin or TBO, which is mostly produced in the liver, and it stimulates the production of your megakaryocytes, and that will lead to the production of more blood platelets. For your white blood cells, the production of white blood cells is typically based on need, and these factors, colony stimulating factor, interleukins, and stem cell factors, can be released to stimulate the production of your white blood cells. These can be produced by your endothelium, by fibroblasts in your bone marrow, and also by other leukocytes or other white blood cells. And this is important because when you have an infection, then your white blood cells will detect this infection and then stimulate the production of even more white blood cells, which will lead to a better immune response to fight off this infection. So let's take a closer look at the formation of your white blood cells. And this process is known as leukopoiesis. So you will learn about the different functions of your leukocytes in your lectures on immunology. But in this lecture we will briefly touch on the formation of the different types of leukocytes. So from your blood stem cell you can see it becomes your myeloid stem cell, which will then form your myeloblast. Now for your monocyte it's slightly different. It also can be known as a monoblast. And this monoblast will become your promonocyte, which will become your monocyte, which will then settle in a specific tissue and become your macrophage. So a macrophage is a matured monocyte. And you can see for your eosinophil, your basophil, and your neutrophil, there are all these other cells in between. So your eosinophilic myelocyte, your basophilic myelocyte, your neutrophilic myelocyte, before it becomes your eosinophil, your basophil, and your neutrophil. It's not important for these lectures for you to know all these cell names in between, but it's just for you to know that there are other cells in between before it matures into your final cell. So before your myeloblast becomes an eosinophil, a basophil, or a neutrophil, there are a few steps in between as well. For your lymphoid stem cell, it can become a B lymphocyte or a T lymphocyte, again depending on the tissue that it matures in. So for your B lymphocyte, it matures in your bone marrow, and your T lymphocyte matures in your thymus. So if we take a closer look at the characteristics of your different types of white blood cells, if you remember, your basophil, your neutrophil, and your eosinophil are granulocytes, whereas your monocytes and your lymphocytes are agranulocytes. Your neutrophils, eosinophils, and monocytes are phagocytes, which means they can phagocytose or ingest pathogens to destroy these pathogens. Your eosinophil and your lymphocytes are cytotoxic cells, and cytotoxic cells means they can destroy your own cells that have been infected with a virus or that have become cancerous. They can destroy these cells before they spread. Your eosinophil and monocytes and lymphocytes can also be antigen presenting cells. So they can present antigens so that your body can produce antibodies against those particular antigens. But again, you will learn about this in more detail in your immunology lectures. If you look at the percentages of your white blood cells, your basophils are quite rare, your neutrophils are the most common, followed by your lymphocytes, and then your monocytes and your eosinophils. And then you get different subtypes of your white blood cells. But again, this will be more clear when you do your immunology lectures. So in this table, you can just see some of the basic functions of your different types of white blood cells, and you'll notice that they have quite unique functions. The cells of myeloid origin tend to play a more important role in your innate immunity, whereas your cells from your lymphoid origin play a more important role in your adaptive immune response. So that's your learned immune response. But again, this will be explained in much more detail in your immunology lectures, so we won't focus on this too much in these lectures. So now let's take a closer look at the formation of your red blood cells. And this process is known as erythropoiesis. So it's the formation of your red blood cells. So erythropoiesis is controlled by the blood oxygen levels, so the amount of oxygen that you have in your blood. And this is because your red blood cells play a very important role in carrying oxygen in your body. So if you have low levels of oxygen in your body, this is known as hypoxia. So hypoxia is a low level of oxygen, and this will be detected by oxygen sensing cells in your kidneys. And when they detect this low level of oxygen, they will then release erythropoietin, or EPO. That will travel to your bone marrow to stimulate the production of more red blood cells. And if you produce more red blood cells, that will increase the oxygen carrying capacity of your blood. So now your blood will be able to carry more oxygen, and that will increase the oxygen levels in your blood and return them back to normal. So this is very important for the homeostatic control of oxygen levels in your body. So as I mentioned, it's the low level of oxygen that will stimulate your kidneys to produce erythropoietin, and erythropoietin will act at the later stages of your stem cell differentiation. So as your stem cell is in a late stage of differentiation, it will also stimulate your pronormoblast to become a reticulocyte, which will then become a circulating red blood cell. So EPO is a glycoprotein, which just means that it's a protein that has some carbohydrate groups attached to it. Most of your EPO as an adult is produced in your kidneys, but a small percentage is produced in the liver as well. In the fetus and in the newborn, most of your EPO gets produced in the liver. The EPO has a half-life of about 5 hours in circulation, but it can increase your red blood cell count for between 2 to 3 days. So even after your EPO levels drop, you can still maintain a high red blood cell production for the next 2 to 3 days. And then once your EPO has completed its job, it gets inactivated in the liver. So we have been able to develop recombinant EPO, and recombinant just means that it's been created in a lab. And this recombinant EPO has been used for many different reasons. It's been used for the treatment of anemia in the case of renal failure. So if you have renal failure, this is when your kidneys fail, and therefore you're not able to produce enough EPO. And because you're not producing enough EPO, then you're not producing enough red blood cells. So you have this decrease in red blood cells, which will lead to anemia, and this can be treated by giving these patients recombinant EPO to increase their red blood cell production. It's also been used to stimulate erythropoiesis for an autologous transfusion. So an autologous transfusion is when you get a transfusion of your own blood. So how this works is before the scheduled surgery, you'll be treated with EPO, the recombinant EPO, and this will cause you to increase your red blood cell production. Your blood will then be collected, and then when it's time for your surgery, you will be transfused with your own blood. This decreases the chance of having a rejection because you're getting your own blood, and because you were treated with EPO, you had a high amount of red blood cells in your blood. So you're going to be transfusing yourself with blood that is rich in red blood cells, and this increases the likelihood of a successful operation. Recombinant EPO has also been used for blood doping. I'm sure we've all heard of the case of Lance Armstrong. He used EPO for blood doping. And this is because it will increase your red blood cell numbers, and that will increase the oxygen carrying capacity of your blood. And because you're carrying a lot more oxygen, this will delay muscle fatigue and help improve your performance. And this is why athletes might want to use recombinant EPO for blood doping to improve their performance. So let's take a closer look at this process of erythropoiesis. So your myeloid stem cell will develop into a proerythroblast, also known as a pronormoblast, which will then develop into an erythroblast, also known as a normoblast. And at this stage, hemoglobin synthesis will begin. So hemoglobin is the oxygen carrying molecule in your red blood cells. Hemoglobin is also a red pigment, and it's what gives your red blood cell its red color, and what will also give your blood its red color. So your erythroblast will eventually kick out the nucleus and get rid of most of the organelles inside the cell. And then it will become this, what's known as a reticulocyte. Your reticulocyte will spend about 3 days in the marrow before it's released in the blood. So your red blood cell is released into the blood as a reticulocyte. So your reticulocyte will still contain a few organelles inside the cell. And then it'll spend about 1 day in the blood as a reticulocyte before it matures into the final erythrocyte or red blood cell. So this is the final mature red blood cell where it contains no organelles and it's just a cell that contains your hemoglobin molecule inside it. So on this slide, I just want to touch on the importance of vitamin B12 and folic acid, which is also known as vitamin B9. And these are two vitamins that are very important for the production of red blood cells. So they work on this very early stage of red blood cell production. So they're very important for the cell division and the maturation of your red blood cells. And without these vitamins, you end up releasing these red blood cells at this very early stage. So these red blood cells are not able to progress into these later stages of maturation. So you end up releasing them into the blood at this very early stage. So you end up having these very large red blood cells that don't contain a lot of hemoglobin and they cannot function properly. And this condition is known as megaloblastic anemia. So megaloblastic because the cells are very large, and anemia because they don't contain a lot of hemoglobin and they don't function properly, so they're not able to carry enough oxygen for the functions of your body. And this is why vitamin B12 and folic acid are very important for red blood cell production. So for vitamin B12 to be absorbed in the body, you need a factor that's known as intrinsic factor. An intrinsic factor is produced by your parietal cells in your stomach. So when you produce this intrinsic factor, it binds to the vitamin B12 that you've ingested, and then your vitamin B12 can be absorbed in the ileum. So you have this absorption of vitamin B12. Vitamin B12 then gets released into the blood, and then it gets transported in the blood by this protein that's known as transcobalamin. So transcobalamin transports vitamin B12 into the blood, and then it takes it to the areas in the body that you need it. So for example, to your bone marrow, where it's then needed for your red blood cell production. So this brings us to the end of the second lecture. In this lecture, we discussed the sites of blood cell production, which differs depending on your stage of life. So it starts off in the yolk sac, and then moves on to the liver, and then eventually your bone marrow takes over as the primary site of blood cell production. We also discussed leukopoiesis, which is the formation of your white blood cells, and erythropoiesis, which is the formation of your red blood cells. We talked about the importance of blood oxygen levels in the formation of your red blood cells, and the importance of EPO, which is very important for stimulating red blood cell production. And we also discussed recombinant EPO and the important functions of recombinant EPO. We mentioned that your red blood cell starts off as a haemopoietic stem cell, which eventually develops into your pro-erythroblast or pro-normoblast, and then your erythroblast or normoblast, and then your reticulocyte, and then finally your mature red blood cell or erythrocyte. And then lastly, we discussed the importance of vitamin B12 and folic acid in your red blood cell production. So this brings us to the end of this lecture. If you have any questions, please use the discussion boards on ClickUp, or you can pop me an email on my email address.